I finished by discussing how you generate the tDNA strand, which is a single-stranded DNA. It's not double-stranded, so you peel away this. Let me move this so I can see because I'm blocking myself. Okay. So you peel away this bottom strand corresponding to the tDNA only. So it's cut here, initially cut at this border, and then here, and then DNA polymerase will initiate synthesis as this is being peeled away. So it contributes, DNA polymerase contributes to removing this strand here. Okay, so you end up with a double-stranded DNA and the plasmid single-stranded DNA attached to BRD2. Okay, so this is just, everybody went through that. So now that we have the question is that now that we have the single-stranded DNA, tDNA, how is it transferred to the plant? And how is that process? How does that process occur? And this involves the VRB operon, which codes for the transfer apparatus. And excuse me. So the proteins that are encoded by the genes in this operon make up the pore through which the tDNA complex is transferred. In a way, this is similar to conjugation. The apparatus is very similar. And these proteins form a channel. I'll show you image of that, a cartoon. And this is going to traverse the inner and outer membranes of the bacterial cell. Now remember, the bacteria are attached to plant cells. Plant cells have cell wall, which is very complex, and also the plasma membrane. Somehow, the DNA has to get through all those layers, but it is not clear how it does it through the plant, how this apparatus interacts with the plant surface. Okay, so here is an image of this apparatus. So this is a type 4 secretion apparatus, which is also used in conjugation. The inner membrane of the bacterial cell, periplasmic space, the outer membrane, and this projects out. As I said, how all of this interacts with the plant cell, the complex cell wall, and the plasma membrane is not clear. But as you see, there are many proteins that are part of this apparatus. Okay, and VRD4, this is not part of the VRB operon, it's part of the VRD operon. This is, part of the operon that encodes VRD2, which is the protein that is going to drag the DNA through this pore. Okay, but this is a very important protein, which is analogous to coupling protein. Okay, so this receptor protein, VRD2, is the one that recognizes VRD2. So they interact, and if they have the appropriate amino acids, then VRD4 will allow this to open up, and VRD2 can go through, and it drags the tDNA, because the tDNA is attached to VRD2. So the DNA is moved passively through this pore. Here is another view, a simplified view of what I showed before, with VRD4 being the gatekeeper, analogous to coupling protein. Okay, so there are 11 genes in this operon. Let me get some water. And 10 of them are essential for transfer, and they encode membrane proteins. As you can see, either they are in the inner membrane or the outer membrane or part of both membranes. And the VRB2 protein codes for pillin, which is the protein of the pilus. That is that extension that sticks out. So this is, again, these proteins are part of a large superfamily that people have found when they compare the genomes of many bacteria, and they are all adapted for the promiscuous transfer of proteins as well as DNA protein complexes, like in the case of tDNA, but that's not the only DNA transfer. Okay. In the case of agrobacterium, what I mean is, in the case of agrobacterium, it's only the tDNA, but there are other interactions of bacteria with other types of cells where you have other DNAs that are being transferred. So the VRE proteins are transferred through this pore, but independently of the tDNA complex. At one point, it was thought that these proteins were part, or they were attached to the tDNA, but now they have shown that that is not the case. But they are, injected into the plant cells through this pore, and they play a critical role in the translation of the tDNA complex to the nucleus of the plant cells. What I mean by that is that their role is in the plant cell. These are bacterial proteins, but they are required when the tDNA is going to be transported from the cytoplasm to the nucleus of the plant cell. These proteins are not required in agrobacterium, the VRE proteins I'm discussing. And VRD and VRE, interestingly, these are bacterial proteins, you have to keep in mind, have nuclear localization signals. There is no nucleus in bacteria, but they have these nuclear localization signals telling you that these proteins play a critical role in localization of the tDNA into the nucleus. And of course, nuclear localization signals are those signals that allow proteins to enter the eukaryotic nucleus. Entry into the eukaryotic nucleus is not a passive phenomenon. You require a lot of proteins because the pores have to expand, right, to allow the entry of proteins and RNAs and other kinds of molecules. Only if the molecules are very small can they diffuse through the nucleus. So the tDNA complex exceeds the size exclusion limit of 40,000 Daltons of the nuclear pore. So that is a limit that people have figured out for most eukaryotic nuclear pores. And so indicating that the entry of tDNA is not passive, that you're going to require a lot of proteins, including chaperones, present in the plant cell to make this entry. And for example, the 20 kb tDNA strand from nopalin TI plasmid has a total mass of 50,000 kilodaltons. So it's huge. It really cannot enter passively. This has to be, involves many proteins, including VRE and VRD with their nuclear localization signals. And this is to show you a schematic of the eukaryotic nuclear pore complex. So here at the bottom is the nuclear side, on the top is the cytoplasmic side, and then these pores normally are quite closed, allow only diffusion of very small molecules, but when larger molecules have to enter, they will open up by the action of many different proteins. Okay, and this is, on the right here is an electron micrograph showing nuclear pores on the nuclear membrane. The rest is fuzzy. I'm not going to go through that. So, one important thing to enter into the nucleus in eukaryotic cells for large molecules is that they have to have a nuclear localization signal. This is for proteins. And these nuclear localization signals interact with proteins that are the part of the import machinery, what brings the proteins through these pores. As I mentioned, VRD2 and VRE2 have nuclear localization signals. And if you delete the nuclear localization signal in VRD2, you reduce but do not abolish nuclear localization. So it says that it's important but not essential because you get some nuclear localization. Now, if you delete the nuclear localization in VRE, you completely abolish nuclear localization. So this experiment shows that VRE2 is really critical in localization of the tDNA, or the nuclear localization in VRE2 is critical for localization of the tDNA to the nucleus. So there are experiments, a lot of very interesting experiments that people have done. For example, if you express VRD2 and VRE2 in the plant cells, they localize in the nucleus. And this is very easy to do. You can just fuse green fluorescent proteins to these proteins. And then you ask, where do they localize? And they localize in the nucleus of the plant cell. This is without agrobacterium, okay? You express them using molecular cloning to express these proteins, just these proteins in the plant cell. There's no bacteria here, there's no infection, etc. And these proteins are localized in the nucleus. This demonstrates that their nuclear localization signals are functional. Okay, because you can have a nuclear localization signal. Just because you have a sequence doesn't mean that it performs that function. Okay, so you have to demonstrate doing the experiment. And this is the experiment that shows that the nuclear localization signal on these proteins is functional and required for them to enter the nucleus. The previous experiment, deletion, shows that you require it. In the case of VRD2, it's absolutely essential. And fusing each of these proteins to GFP and localization to the nucleus demonstrates that their nuclear localization signal is functional. So a lot of other experiments were done to understand the role of VRE, because at one point, as I mentioned, it was very controversial. They didn't know if it was playing a role with the tDNA complex in transport across the type 4 secretion apparatus. But if you, for example, have the single-stranded DNA alone, meaning tDNA, there's no nuclear localization. If you add the tDNA with VRE, these are experiments done in planta, so in the plant only, no bacteria here. So if you express the tDNA, and there are ways to do that, and VRE, you have nuclear localization. Now if you do a co-infection, now in this experiment you will have a co-infection, then you have nuclear and cytoplasmic localization. Okay, so, and I cannot see here. So VRE really is mediating, is the major player in moving the tDNA into the nucleus of the host cell. Get rid of this. Okay, so there are other issues that arise in understanding this system. One of them is that the plant hormones, remember the tDNA has plant hormones, and these hormones are not expressed in the bacterium. They are only expressed in the plant cell, but they are carried on DNA that is present in the bacterium. So if they are to be expressed in the plant cell, they must have signals, regulatory signals that are recognized by the transcriptional machinery and the translational machinery to be expressed in plant cells.